So we know that like for the past decades, we have seen like a lot of phylogenies have been reconstructed for basically all organisms on Earth. So here I just picked two examples, like in spiders and cacti. So we now have like a lot of molecular sequencing, a lot of capacity for gathering data and to evaluate with modern computers. And we are getting more and more of these, and we have a lot of sections in this Congress talking about that. And my point is, like, with all these phylogenies, what we can do is now address a lot of the questions on morphological evolution using this knowledge on the hypothesis of relationship of species. So just to give an example, here we have, like, to give examples outside of wood anatomy. So for example, in the evolution of tendrils in lianas of Ignomniaceae, we have here, for example, from leaves that have tendrils that have like three terminations, trifid tendrils giving rise to like simple tendrils or multifid tendrils in other species. So basically my point is that now having the phylogenies, we are able to reevaluate all the diversity that we have in plant and animals and all living organisms on Earth. And so where does we stand like in bark anatomy and wood anatomy in the present? So we are all celebrating the Irving Bailey here today in the symposium, and so the basis of what we know about evolution, of what we think evolution was like in xylem, was based in the idea that from tracheids we had the evolution of fibers and vessels, and the word specialization will appear. So with that concept of evolution as a ladder of progress that is pervasive in most of like animal or plant evolution in all the areas of organismal evolution, and the idea that because evolution was a synonym of progress, that it would go in one direction. So here from perforation plates that were tend to be thought as not as efficient in conduction to others that were more efficient. And of course, Bailey also was aware that you like the word potentialities, but also limitations to like the use of wood anatomy to understand evolution. For example, he was aware of convergent parallel evolution. So he says back then that we should not establish relationships in plants solely from wood anatomy, but we should also use other influences as well. And nowadays, that's the main like basis of what we do. We try to gather a lot of data from genes or from morphology to reconstruct better like the idea of relationships in organisms. But still, like the main, and it was not only Bailey, but everybody in that moment is thinking like in evolution as a progress. So he says that we can use wood anatomy to negate hypotheses of ancestry. So a plant with a scalariform perforation plate would not be derived from an ancestor with simple perforation plate. And the same ideas of xylem evolution goes to phloem evolution. So you see Hemingway here in 1913. So that this idea, this period is flourishing with these ideas of evolution in different parts of the plants and animals. And also the same is proposed, like from a sieve cell to a sieve tube, the same trend would be expected. But today we know, as Elizabeth has mentioned, and a lot of us will mention, that this is not the case. And in many, although you have in general lines, we know that tracheids appeared first. So we know that the ancestor of tracheophytes had tracheids and not vessels. So we know that a lot of these trends are in fact things that happen in evolution, but we know that evolution is not unidirectional. So here we have in the magnoliads the evolution of bearing woods only with only tracheids, like in the Winteraceae or also in the Eudicots. In Trocodendrales, you have like plants with exclusively tracheids. So showing us that evolution is not like unidirectional. And the same can be seen in numerous other organisms, like even in Araliaceae, Alexei Oskowski saw like ancestors probably with simple perforation plates giving rise to organ like other species with scalariform perforation plates. So, but what can we learn then from a tree? So there are lots of things that we can do with the phylogenies nowadays, like from ancestral characteristics reconstruction to analysis of diversification, correlated evolution. And what I want to do is show some of these results that I have for two plant families that I have been studying. my main interest is to understand the evolution of complex types of anatomies, but today I'll be focusing exclusively in other of my line of research that is trying to study wood and bark evolution. And of course, just to give an idea, so I will talk mainly about Bignoniaceae. So they are a tropical family. So with pantropical distribution, there are a lot of lianas in the families. In fact, like half of the number of species in the family are of lianas, but also a lot of important trees, also economically important trees, as the Androanthus that have very important woods, that we'll talk a little bit more in future. Of course, there are limitations, because I'll be talking of a family within the Lamiales. I won't have much of a diversity in the tracheary elements. So I'll be focusing most of my presentation in the evolution of other types of cells from the secondary xylem. And I'm thankful that Elizabeth already said, like a lot of this, what was proposed as the line of evolution for these types of cells. So for ray evolution, the idea was going from heterocellular arrays to homocellular arrays with procumbent cells. And for axial parenchyma, this is a diagram done by Kalmqvist. And so from diffuse to forms like of paratracheal parenchyma, aliform complex. So that's the idea of the cribs and frogs, all of them have posed back in that moment. And in Bignoniaceae, first of all, I would like to mention that there has been a lot of recent phylogenetic work in the family by Richard Olmsted, by Lucia Lohmann, and Michel Zaira. So what we know, Bignoniaceae was quite heterogeneous, but now with the new phylogenies, we see that wood anatomically it's very, very consistent. Like you can delimit groups and have synapomorphism for these groups based in wood anatomy. Some of these clades are exclusively like, when they were reconstructed, they had no morphological synapomorphy, so it's nice to see that we can find some evidence of morphology and anatomy that sustain that. And one of these examples I'll give, this is Androanthus, this is a lamppost, a light post, and it resprouted. So this is Brazilian national tree, and it was shown to be polyphyletic. So we have a lot of lineages, at least appears in 3 lineages within this clade of the Neotropics. And the interesting thing is that this genus in wood anatomy was known to be very diverse, and it matched exactly what wood anatomy was known. For example, the hardwoods of, we always knew that there were some of the tabebuyas were really heavy woods and really valuable as a timber, and some were very light and used as boxes. We call it caixeta in Portuguese because it's used like a caixa, it's a box. And in fact, like one of the clades, this clade corresponds to Recording-Hess Lapacho group. So it's where these substances in the vessels, a yellowish substance, very dense woods, a more marked storage structure, and very wide intravessel pits. The other big group corresponds exactly to the group without lepacol and with light. It can be also some of high density, but all the light tabebuyas are here with sometimes more irregularly stored structure and narrow intravessel pits. And the only species that came the Garciala dos Santos and Regis Miller could not fit in the group was the one that actually was not related to any of these 2 clades, which is now known as Rosodendron. They have the widest raising tabebuya and parenchyma strands with a lot of cells. Also, in the Paleotropics, we have a central diversity of Bignoniaceae in Madagascar, and we see that in Madagascar they are the only Bignoniaceae with apotracheal parenchyma. There is paratracheal parenchyma, but also apotracheal parenchyma, which is interesting. Raising Bignoniaceae, they vary a lot in composition. They can be homocellular or heterocellular. And when we see in the phylogeny, we see the evolution going in 2 directions. We see in some lineages the evolution from an ancestor that is reconstructed as homocellular to heterocellular, to lineages exclusively homocellular, but we also see the opposite happening from not so heterocellular rays to very heterocellular rays. And when we see that in confront with the phylogeny of the habits, we see that in the evolution of lianas, most of them get heterocellular arrays with more erect, upright cells. And maybe Kaisa, I let you know, but like Pace has hypothesized that probably like in plants that have narrow stems, the upright and square cells will be acting more in transport vertically and not so much horizontally. And that's what we see in both shrubs and lianas in Bignoniaceae. And so another aspect that also is very varied in Bignoniaceae is the braid width and height. And that also is seen in direct correlation with the evolution of lianas. So in the evolution of habit in Bignoniaceae, a lot of the wood characteristics change, and that has been actually coined as the lianescent vascular syndrome. So if you look at any liana, you will see similar trends, like similar things. So here in a ficus that is a crambler or a liana, you see the ray height in comparison to an arboreal ficus. And what about the phloem? So phloem evolution, so that idea that from compound sieve plate we would go to simple sieve plate, at least in Bignoniaceae that's not what happens. Although when you just plot compound versus simple, you do see that there's evolution of simple from lineages with compound sieve plates. But when you look in detail, like what is the number of sieve areas per sieve plate, you see that in some lineages, they reduce in number, while in others they increase in number. So here to see a little bit better, so here you have like less sieve areas per sieve plate, and here you have up to 40 sieve areas per sieve plate. And there's always something that happens when the sieve plate is simple, the sieve elements were short, and when the sieve plates were compound, the sieve elements were long. That's something that Izu has already mentioned in her 1969 book. And what we saw happening in correlation with that, it's always in the lands with compound sieve plates, a lot of fibers were present. And in those with simple sieve plates, the much less fibers were present. So there was a correlated evolution. The fibers, and if you look in other plants, like in Ugalandaceae, you also have phloems with a lot of fibers, and also the sieve tube members are long and with compound sieve areas. And what we have been looking at, so in Bignoniaceae you see two very opposite types of phloems. And recently we have been doing some calculations in the Amazon forest with some collaborators in Brazil. And they have been calculating the diametral growth of lianas in the forest in the past 10 years. And curiously, we're working with Johannes Lisch, that works with phloem physiology, to try to understand, because what we saw is that the plants with compound sieve plates are the ones that grow faster than the ones that have simple sieve plates. So there might be something telling that, and also we imagine that more fibers also sustain long sieve tube element, but that's still something that is ongoing. And so having phylogenies in hands, I think we could go on and look in a lot of families, what's going on in terms of what features also promote or may be related to differences in diversification rates, to try to understand adaptation in a lot of these aspects. And I would like to thank you and all of these institutions. This is Veronica analyzing one of the matrices that we did. So she couldn't be here, but I'm sure she would like to be here. And thank you very much.